So it turns out there's another whole domain with it, which is these proteins can become soluble, which means they're no longer anchored to a cell, they're just floating around. And ultimately, they're floating around in your saliva, in your urine, in your armpit exudates, or whatever it is. And what they begin to do, complicated mechanisms, which I think I will bypass, what they do is give a unique signature to the pheromones coming off of you. And as we will see in lectures to come, animals of all sorts of species can tell, is this individual of my species? Are they the same gender? Or are they an adult? Are they sexually mature? Or are they healthy? Or are they pregnant? Whatever. But thanks to this major histocompatibility business, they can also tell, is this a relative? Now, the juggling events, the splicing and the juggling of the genes, has some degree of statistical relatedness the more closely related you are. In other words, you smell your own urine, if that's your hobby, and the major histocompatibility genes in there will obviously exactly match your own. You smell those of a full sibling, even more questions to be asked, and you do that, and there will be a greater degree, statistically, of similarity of the structure of that protein than with a second cousin, than with a perfect stranger, than with a Nepalese if you're from Belgium, whatever it is. What you find in those cases is that's how you not only get innate olfactory recognition of is this a relative or not, but how related of a relative. So that's half of it. That's how you generate the unique signature at the olfaction, at the pheromone end. The other trick is, how do you generate an olfactory system that could make that discrimination? And all we got to go with at that point is, you can guess, if you think about it a bit, is we got to have some version of olfactory receptors that do the old lock and key business. Just as you make a protein, which has a certain shape, indicating that this is your unique signature. What you want to do is have receptors that will have the uniquely complementary shape for the lock and key, so that you can do oh, this fits perfectly. Let's transduce this to a signal to the brain saying, I'm smelling my armpit. And if instead you've got a protein that fits in there like a lock and key, but not quite as well, you send the message of, oh, I'm smelling my sibling's armpit. And if it fits in there not quite as well, and all the way down, you could begin to see exactly how you designed this. If you've got 1,000 of these receptors of this shape, and every single one of them, it has them fitting in well enough to stay in there for 3 seconds, so that all 1,000 of them send the signal. It takes 3 seconds of binding there to cause the signal to happen. All 1,000 of them send the signal. It means it's me. It doesn't fit quite as well, so statistically only 800 of them stay in there for 3 seconds. So 800 of the cells are reporting, oh, that's a full sibling. I don't know if that's the mechanism, but this would be a way of constructing it. That's exactly how it could be, along the lines of lock and key, genes produce proteins of certain shapes, certain functions, all of that, and that's how you begin to do it. What have we just gotten? We've gotten a protein, a molecular basis of our theorizing the other day of an if-then clause. If and only if this is a close relative, then send the message to the neurons that do cooperation. We already know that's gibberish to say that there's neurons that do cooperation, but you could begin to see how this is going to work. This is the if part of all the conditional if-then clauses built around degree of relatedness. So how does your olfactory bulb do this? Very interestingly, people are beginning to get a sense of 2 hormones that are relevant to this. One is called oxytocin, and the other is called vasopressin. And what we will see in lectures to come is, particularly in females, oxytocin has long been known to play some like plumbing nuts and bolts job in giving birth, and vasopressin and uterine contractions, and take your average like off the rack endocrinologist, and what these hormones are about is like your uterus contracting and giving birth. But oh, that's so little of what they do. What they're much more interestingly involved with is what happens next, which is now beginning to learn how to recognize the smell of the individual you just gave birth to. Because it turns out what oxytocin and vasopressin do in the olfactory bulbs, the olfactory system, the olfactory equivalent of your eyes and ears, is they tune up the cells that recognize major histocompatibility signals. They make them attuned to, is this a relative or not? And there's an if-then clause. If the levels of this hormone have the certain high levels indicating that I just gave birth, and I smell something whose signature odorants fit really well into this receptor, then this is someone who I'm going to nurse like crazy unless it turns out to be my mother or grandmother. Okay, let's put another. If they're very little and cutesy and make cute little mewling sounds, then I will take care of them and nurse them and all that sort of thing. And it's turning out that that's what oxytocin and vasopressin are doing there. You generate mice with genetically knocking out oxytocin or vasopressin, or their receptors. If these are totally new terms, this will come by the week after next as an introduction. You knock out those genes and you get what is called a social anosmia. Anosmia is the inability to smell something. A social anosmia is your nose is working just fine. If you're a rat, you can discriminate different food types, completely arbitrary odors. You just can't distinguish between individuals. And there was a paper a couple of weeks ago showing for the first time what the model has always been is that circulating oxytocin and vasopressin get into your olfactory system and have those effects. What this paper showed for the first time is you're making those hormones right in your nostrils to begin with. This is what tunes it up. Very interestingly, there's also a literature emerging suggesting mutations in genes relevant to oxytocin and vasopressin in families with a high incidence of autism. Autism, a disease where one way of characterizing it is there are enormous deficits in normal socialization, interaction, social bonding, social affiliation, and this suggests that has something to do with it. Okay, so that's a first mechanism.